All right, so today we're going to be talking about clinical trials, and we're going to start off talking about what a clinical trial is and why I should care. We'll quickly go over some historical examples. We'll talk about the types of study questions that can be addressed by clinical trials, talk a little bit about patient recruitment and some particular difficulties that can happen there, the specific concerns with data collection and management. We'll end up talking about what you end up with a clinical trial, and then very briefly go over steps in launching a clinical trial if this is something that you're interested in doing yourself. So to start off, a clinical trial is a research study with a specific purpose to evaluate a medical, surgical, or behavioral intervention. This is the typical way that researchers discover whether a new treatment, like a new drug, a diet, a medical device, a diagnostic procedure, is safe and effective in people. The NIH also has a definition for clinical trials, and they define it as a research study in which one or more human subjects are prospectively assigned to one or more interventions, that can include a placebo or a control, to evaluate the effects of those interventions on health-related biomedical or behavioral outcomes. Now, it's important to note that the NIH's definition of a clinical trial is more broad and inclusive than what is typically thought of. And this change came about a number of years ago where NIH decided that any study that's involving human subjects, where they're prospectively assigned to conditions and could have an impact on some form of health outcome, should be a clinical trial. So this opened up what NIH considers a clinical trial to many more types of studies than were previously done. Now, clinical trials can be used to answer a number of questions. Again, perhaps most common is looking at the effectiveness of an intervention to treat a disease. So I've come up with some new drug, and I want to see if it has an impact on this condition that I've been studying. As far as an early example goes, one of, if not the earliest, actually was performed in 1747. And the real hallmark of this study and what places it as one of the forerunners to a modern day clinical trial is the fact that there was a controlled clinical trial condition. So the study was examining scurvy, and you may or may not know that the symptoms with scurvy are bleeding gums, weakness, slow wound healing, and they started seeing this a lot more amongst sailors. So James Lind in 1747 decided, well, I'm going to look into what's happening with these individuals on a boat, and conducted this study on the HMS Salisbury in, like I said, 1747. So he took 12 sailors that were showing these symptoms that had scurvy and split them into 6 pairs. And each pair was provided a different dietary supplement. So James Lind previously had decided, oh, maybe diet has something to do with this condition because we don't see it when people are inland as often. So maybe it's something specific about being in this unique environment, being on a boat. And one of the things that goes along with long sea travel is difficulty in bringing along a wide variety of food sources. So we thought maybe it's the diet of the individuals that is resulting in this condition. So he came up with different supplements to add to their diet and then wanted to see what effect they had. So cider, sulfuric acid, vinegar, seawater, a concoction of garlic, mustard seed, balsam of Peru, and myrrh, and 2 oranges and a lemon. And what he saw was that sailors that received the citrus fruit showed improvement compared to other groups. So what he was able to show here via the use of these control conditions of these different dietary supplements, that those that were receiving the citrus fruit showed improvement, which allows James Lind to say, well, maybe it's the presence of the citrus fruit that is actually having an positive effect in this case. So again, the groundbreaking aspects of this study, use of control groups, and then a systematic approach to examining the issue. And in fact, following this study, the British Navy adopted lemon juice as a standard issue. So not only were they convinced that this actually may be what the cause of scurvy is, but they were so much so convinced that they now required all Navy ships to bring along citrus supplement to prevent this from occurring within their sailors. So that's sort of a good example of a clinical study. You know, we were off on the right foot when we started. Unfortunately, it wasn't too long afterwards that people started doing things utilizing clinical studies that really should not have been done. So what we can think of as some early horror stories. And the first one that we'll briefly talk about is the Tuskegee experiments in 1932. So these were conducted by the US Public Health Service and Tuskegee Institute in Alabama. And the purpose of the study was to record progression of syphilis within African American populations. So this was a known issue in certain populations that the US Public Health Service wanted to study and to track, which in itself is not necessarily a bad thing. However, as we'll see, the way that they did it certainly wouldn't pass an IRB board today, and is certainly considered highly unethical. So they had 600 participants, 399 with syphilis, 201 without. And when they were taking part in this study, what they were told is that they were being treated for bad blood in quotation marks, because there is no such thing. So they just made up this condition of bad blood and said, this is what we're treating you for. They did not receive the treatment needed even after penicillin became standard care. So the Tuskegee experiments straddled the time period where we didn't have a great treatment for syphilis, and then we did. In this specific case, even once it was known, hey, if you have syphilis, this penicillin treatment will be able to effectively cure it, they were not offered that treatment. So that's certainly one problem with this study. One of the things that we always want to think about with clinical trials is, well, what do we do if it turns out that our treatment is actually really effective? You know, when we're designing these, we'll have a control group that may be getting standard of care. So these people are just getting what they would typically get if your treatment didn't exist. If it turns out that your treatment is successful, if we learn, hey, this is something that is going to help this, what you should do, and what IRBs are going to be looking for you to do, is after the completion of your study, after people have gone through, if you show that there's a benefit, you offer that to those that may have been in a control condition, so that we're receiving some sort of placebo treatment or a traditional standard of care. You can say, hey, our research has shown that this is actually far more effective. Do you want to do this instead? It turns out you were in the control condition. So offer it to those individuals that may have been randomly assigned to not receive this newfangled treatment, now get the opportunity to. Other ethical breaches here were a lack of informed consent. So as we'll be talking about, one of the pillars of a clinical trial is this notion of informed consent, that you provide your research participants enough information to make an informed decision as to whether this is something that they want to participate in. In this case, that wasn't done at all. Participants were not really told what they are being studied for. They weren't really given an option. They were just said, hey, we're going to treat you for bad blood. And that was considered enrolling them in this study. As I had mentioned a few moments ago, there was an entire lack of treatment following, once it was identified, hey, penicillin is going to resolve these symptoms, they weren't offered it. So that is definitely an ethical breach. There were non-transparent and deceptive practices throughout the experiment. So people that were participating through did not know what the purpose of the study was, what they were doing there. And oftentimes information was actually hidden from the participants to have them keep going through. If they knew, hey, you know, there's this newfangled treatment, we're not going to offer it to you, but you may want to look into that. They weren't even told that. They were just told that you're being treated for bad blood. There were also coercive payments for participation in this study. So participants were told that they get free medical exams, they'd get food, and they'd get burial insurance if they weren't able to survive. And when you're dealing with lower socioeconomic status groups, this is something that you really want to pay attention to. So another thing that IRBs are going to be looking at is whether the payments that individuals are receiving for the study are viewed as unduly coercive. So is the payment or the incentives for participating in the study so high that someone that wouldn't want to do it will do it to get that payment, or to get, in this case, those services? You know, hey, I get free medical exams that I can't afford, I'll get some food, and I get burial insurance if I participate. In this group, that would have been viewed as coercive. Now, how you determine what amount is coercive is going to be difficult, and it's going to be dependent on the specifics of your study population. What's coercive in one group may not be in another, and that's perfectly okay. That's the job of the individuals that are creating the experiment, the clinical trial, and those that are reviewing it from an IRB standpoint, from a regulatory standpoint, to see if that really is the case or not. So for example, when we run studies in our lab, we're working with oftentimes undergraduate students. Paying them $20, $30 an hour is not unduly coercive. If they want to participate, they can get that money. It's a great incentive for doing some of the work that we require them to do, but it's not so much that they would do it if they didn't feel like they should. So that type of incentive is perfectly fine. If we were dealing with participants that were of severe lower socioeconomic status, maybe that amount would be viewed as coercive, and that's something to keep in mind. Unfortunately, the horror stories don't end there. So another example is the Nuremberg doctor trials that took place following World War II. So I'm sure many of you are aware that during World War II, especially within concentration camps, there was a fair bit of medical experimentation going on that had zero consent from those that were participating. These were tests looking at how the body responded to high altitudes as the Germany was conducting, or its Air Force was expanding into things like jet propulsion and stuff. They wanted to know how high can the human body sort of survive. So they were doing studies with these completely non-consensual research participants, in quotation marks, to see at what level the body starts to suffer negative consequences for being at high altitudes. Similar with freezing temperatures. At what point does the human body start to suffer consequences from being in lower temperatures? There was a lot of testing of drugs and treatments. So just seeing what this dosage of this pharmacological agent does to the body. So definitely a low point in clinical trials, and some horrific things were done during that time, but it did result in what we now refer to as the Nuremberg Code, which was one of the first steps in really putting into practice laws to prevent this sort of stuff from happening in the future. In the 50s and 60s, there was the thalidomide tragedy. So thalidomide was used as a sedative in the 50s and found to be effective for morning sickness. So this drug was well known to be useful as a sedative, and it turns out that individuals that were taking it whilst they were pregnant didn't experience morning sickness. So that sounded like a great thing. Hey, we can prescribe this sedative to individuals and their morning sickness will go away if they're pregnant. However, a few years later, primarily in the 60s, it actually became evident that thalidomide was causing specific types of birth defects, so shortened or absent limbs in the babies of people that were taking this during pregnancy. So this case resulted in the FDA actually being empowered, so it received stronger authority over drug regulations, known as something called this drug amendment here. So in order to move a drug into clinical trials, especially later clinical trials, you have to get FDA approval. For that FDA approval, you have to do things like prove that there is effectiveness and safety of the drug, first in animal models typically, and then in human models. And then once that's done, you can start to do those larger stage clinical trials and ultimately roll out this treatment to the market. You have to disclose side effects and efficacy. So it's not enough just to say that there may be side effects. I'm sure you've all seen those commercials where you're treating for something like erectile dysfunction. At the end, they say all of these horrible things that can happen that make you wonder whether taking the drug is worth it or not. The reason why they do that is the after effects of the thalidomide tragedy. So it's now in important for and required for drug companies to say all of the side effects that are being associated with taking this specific drug. And then, of course, informed consent for participating in clinical trials was also brought in under this time. So I mentioned a few moments ago about this idea of this staggered approach to clinical trials, different phases of clinical trials. And I'm sure we've all heard the term such and such is going into a phase 3 clinical trial or is currently at phase 2 clinical trial. What do we mean by those? So a phase 1 clinical trial is typically the first in humans, and the purpose is often to find a safe dose. So this is done with smaller numbers of participants, typically 15 to 30, again, really dependent on the specifics of the drug that you're studying and the number of people that your power analysis say are required to see a difference here. But it's really just to find out how safe this medication is in humans. Of course, this will take place following animal work, typically. Phase 2 is typically designed to determine efficacy. So once you find that a specific dose seems to be tolerable, to be safe, what the side effects are, then you can move on to a phase 2 trial where you want to look at whether it has any impact on the thing that you're actually studying. These typically have less than 100 participants, but again, it's really going to be dependent on the specifics of the treatment that you're examining. And then following phase 2, if everything goes well and you show that this new treatment or this new diagnostic technique has merit, it has some usefulness to it, then you can compare this new thing to clinical standards. So compared to what's done previously. And this is going to have hundreds of participants, often in the thousands to hundreds of thousands. And now you're trying to show that this new method is better than what's currently out there. Following that, we have phase 4 clinical trials, and these actually occur post-market. So these typically occur after a drug or a treatment or a diagnostic procedure has made it through all of these first 3 phases to great effect. It's shown to be useful, and then following that, once it's approved as a drug or a treatment by the FDA, it's still monitored in this phase 4, where you're looking at long-term safety and efficacy. And this will have, again, hundreds to thousands of participants, depending on how many people are taking the drug that you are creating. Now, one of the things that's different about clinical trials than basic science research or those types of questions is that they require more resources than non-clinical trial studies. So the types of resources, if you just think about what's required to run a clinical trial, obviously you need patients that have the condition that you're interested in studying or that you are interested in improving diagnosis. So you require that, but then you also need control participants of one sort or another. So already you have typically larger studies than may be done otherwise. Physicians are, of course, going to be highly involved in this type of study. Typically, they're the ones that can assess the clinical utility. They are oftentimes the ones that are able to prescribe whatever it is that you're doing, and they are the ones that have the access to the clinical populations, and of course, a deep understanding of the clinical condition. So oftentimes the investigator that has created a potential treatment has done this based on basic science leading up to it. They may not be a clinical expert at all. So it's important to have physicians on the team to be able to say, how this may actually play out in the real world with patients, and that's going to be required. Nursing staff, similarly, pharmacists, oftentimes when you are creating a drug, you need to have a compounding lab involved. So someone to actually package the drugs in a way that meet all of the guidelines for a clinical trial. So for example, the placebo versus the experimental condition should be as comparable as possible. And by that, what I mean is you can't have one in pill form and the other one being a liquid. You want them both to be in pill form, and oftentimes this requires the involvement of pharmacists to actually compound the placebo and the experimental treatment in a way that they're as similar as possible. Lab techs, data managers, a whole team is formed around a clinical trial. Some of these members will be within a university setting typically, and some of them will be within a hospital setting, and these areas have to talk to one another a great deal. Now, as far as the types of study questions, I've sort of introduced many of these already, but you may want to look at treatment or therapy. So you have a novel way to treat a disease, and you want to see if it's going to be useful. Second, prevention. So if you come up with a way to prevent a disease, perhaps it's a prophylactic treatment, perhaps it's some sort of environmental treatment or exposure that you've identified that can be preventative, that's also going to require a clinical trial. Diagnostic tests. Perhaps your treatment has nothing to do, or your study has nothing to do with treating a disease, but instead you're interested in coming up with improved ways to diagnose it. A lot of conditions may not have a gold standard diagnostic technique. Those also require clinical trials to move forward. So this blood assay may show an increase in the ability to identify a condition, or this radiological procedure. Hey, when I do this type of exam, I'm more likely to be able to correctly diagnose this condition that's difficult to see otherwise. All require clinical trials. Same thing with early detection or screening. I have a new way to find a hidden disease. So something that is certainly in line with diagnostic tests, but a little bit different conceptually. Now, the design of clinical trials is something that you need to start thinking about pretty much straight away if this is something that you're interested in doing. So of course, randomization is going to be an important part here, and we saw this back in the scurvy study in 1747. How participants are going to be assigned to conditions, you have to figure out how to perform that randomization. You don't want to have any systematic factor used in the assigning of conditions, because then you may have some spurious effect that you need to be aware of that you are missing. So a good example of this would be, if the first 5 people that sign up for the study, you put in the treatment condition, and the next 5 you put in the control condition, well, what you've done now is confounded their eagerness to participate. Maybe the first 5 people, because of their eagerness, are more likely to stick to the treatment, or are more likely to follow directions, or the general compliance sorts of questions than the second group. So if you are looking at comparing these 2 groups, there's already something that's inherently different about them that could influence your results. So it's important to perform randomization so every participant has an equal likelihood of appearing in one of the conditions, whether it be a control condition or an experimental condition. Blinding is another important component of clinical trials that we often are somewhat aware of, but really want to pay a lot of conscious thought to, and specifically the type of blinding that you're going to be doing. So you definitely want to have, at a minimum, your study to be single-blinded. And when we say single-blinded, what we mean is the person who's participating doesn't know what condition they are in. If they know what condition they're in, now you are very likely to show placebo effects, and they're going to muck around with your data, and it's going to make it very difficult to interpret. So at a minimum, you want your research participant to not know whether they are in a control condition or in the experimental condition. Ideally, we would also like the person interacting with the participant to be blind as to whether they're in the control condition or the experimental condition. And the reason for this is it may change the way that they are interacted with. So if you know that the person that just showed up in your office to receive this treatment is in the control condition and they're not actually receiving an experimental medication, that could influence how you behave around them. That could influence how you interpret the things that they say. So if they report, oh yeah, I had this side effect, you may start thinking that, oh, well, it can't possibly have anything to do with what I am giving them, because it clearly isn't a result of the placebo. So you may not record that. Those sorts of differences can be important as well. So you want to make sure, or ideally, the person that's interacting with the participant also doesn't know what condition they're in. And then A triple blinded study is where the participant has no idea what condition they're in, the investigator that's interacting with the participant has no idea what condition they're in, and the person who's looking at the data and performing the analyses has no idea what condition they're in. And this is sort of another level of blinding that provides even further confidence in what you're seeing. So in these cases, what you are doing is you're preventing a not necessarily intentional way of, for example, data manipulation, but even unintentional sorts of things. If there's any sort of subjectivity in coding data, you certainly wouldn't want the person that is doing the data analysis to know what condition people are in. And of course, this does eliminate the possibility of intentional data manipulation that's going to stop you from, or that is going to influence results. So a triple blinded study, the investigator, the research participant, and the person doing the analysis doesn't know what groups people are in. And that, I would say, is sort of the gold standard. But what we see most often is a double blinded study, and a single blinded study nowadays probably isn't enough. You want double at a minimum. We've already talked about control groups, and the number of control groups can vary from study to study, but it's important for you to have some sort of standard for comparison. So how do you know what you're doing is having an impact that isn't just random? You need to have a group that has received everything as similarly to the actual study as possible, with the exception of the treatment. So if it is a pharmacological agent that they're taking, you want them to have to take a pill just like your experimental group at the same frequency, the same time, it looks the same, it comes in a similar bottle. You want all of that stuff matched, and then any effect that you see between our experimental group and our control group is a result of the medication, not of just things like expectancy biases or placebo effects. So in line with control groups, you do need a placebo condition, and this could be a number of different things, but the most clear cut is in a drug trial where your placebo treatment is essentially a sugar pill compared to one that actually has active ingredients within it. Now, we've already talked about some of the ethical considerations in clinical trials, so we'll return to this quite briefly, but one of the fundamental components of a clinical trial is this idea of informed consent. Again, ensuring participants understand the risks and benefits so they can decide if this is something that they want to do. So typically, an informed consent form will talk about the purpose of the study, the risks and benefits, any potential alternative treatments. So if you don't do this, what options are there? How are your records going to be maintained, confidentiality-wise? Who's going to have access to this? Is it going to end up in your health insurance records, or is it something that just stays with the study team? Who can see this? Who's going to pay for medical treatment if injury occurs as you're participating? What if something bad happens? Is that up to the study team, or is that up to you to provide that treatment? Who do you contact if you have questions? What if you're going to be late? Who do you talk to in those cases? And of course, highlighting to the participant as much as is necessary that this is going to be voluntary. You can decide that you want to start now, and if 5 minutes in you say you don't want to participate anymore, that's fine. And that it's not going to impact your care other than through participation in the study. So at any point, it's essential that a patient realizes and is aware that they can withdraw their consent. Just because you signed on day one doesn't mean that you have to keep going. Maybe the side effects are something that you don't want to tolerate. You can withdraw. Maybe you thought that it'd be more interesting than it is, or more fun. Whatever your reasons for participating are yours, that's perfectly fine. Likewise, your reasons to withdraw from the study are also yours and perfectly fine. You will never be coerced to continue a study if it's a clinical trial that's being conducted ethically. Of course, we've talked about the role of institutional review boards in previous lectures and in previous slides in this lecture, but it's important that an IRB is approving a study. That's going to ensure that the clinical trial is ethical, that it's meeting all of these requirements, and it's also a group that looks out for the rights and welfare of the study participants and make sure that they're protected. So like we talked about in the module about institutional review boards, this will be a group of pretty well reasoned, pretty well informed individuals in a combination of medical and academic settings that are reviewing these studies and determining whether there's the potential for benefit, whether all of these safeguards and policies are in place, whether the information that's presented to participants is enough to ensure that they can provide their informed consent. They're watching out for all of that. They're also reviewing these studies every 6 months, every 12 months, depending on the relative risk, and seeing how things are going and making corrections if need be. In a similar line, there's oftentimes, if not always, in a clinical study, a patient safety monitoring board. So this is going to be typically an independent group that's not related to the study, that is reviewing the medical aspects of the study, the medical outcomes, and determining whether it's safe for participants to participate in, and whether the outcomes that are being reported are beneficial. So they're an educated group, a group that's educated medically, that is looking at the outcomes of the study. So some of the additional challenges with clinical trials, apart from all of the ethical considerations that are quite onerous, important, but a lot of work, are perhaps the most difficult is recruitment and retention. It is typically the number reason why clinical trials fail. So not only do you have to identify participants and patients to participate in this study, which in itself can be a fairly arduous task, a difficult thing to do, especially depending on what condition you're looking at treating. You also have to have them complete the study protocol. So clinical trials are oftentimes quite long. They require multiple follow-ups. You can't have your patient provide points sort of sporadically to get a good picture. You need to have them followed through the entire duration of the clinical trial, and that can be something that can be problematic or difficult to attain. Again, participants, it's exceptionally important that they have the right to withdraw whenever they feel like it. And as a study designer, as the person that's conducting the research, you want to make sure that you have things in place that minimizes that occurrence, not through coercion, not through deception, but by engaging in common practices that are viewed to be the best sort of ways to have participants go through clinical studies. So some strategies are, firstly, physician trust. It's exceptionally important for there to be a bond between the physician that is looking at enrolling individuals in the study and the individuals themselves. Again, this cannot be a coercive type of bond. It can't be, hey, I'm your doctor, I know what's best for you, you should totally do this. It has to be at a level of respect from the patient and their medical provider, thinking, well, they often do what is in my best interest. So if they're recommending this, this also may be in my best interest, and it's something that I may want to look at. And we see this a lot in populations where there isn't that level of trust. Participation rates are much, much lower. So anything that you can do to garner that trust with your potential research participants is very important there. Of course, you need to have adequate study staff. So you need people that are able to check in with your patients, make sure that they're aware of upcoming appointments, changes in schedules as need be. This can't be something that one person is doing on their own. You'll have people fall through the cracks. You'll have changes that need to be instituted that can't be, because you just don't have the personnel to do so. Some ways to garner or to generate recruitment are by doing things like putting out newspaper and radio ads. I'm sure you've all seen on like Facebook feeds and things that a certain research group is recruiting for a trial. So getting the word out as broadly as possible. You can also create websites and things. And then of course, lastly, physician referrals. So if you're not either in a position to interact with patients, or it's deemed that it could be coercive if you're interacting with your patients directly, having other physicians that aren't involved in the study recommending or providing information about this research opportunity is going to be important. So getting the word out there for you. Another problem with clinical trials is the funding. So clinical trials are exceptionally expensive. Oftentimes, to go from a phase 1 to a phase 4 clinical trial costs tens of millions of dollars. And this is something that you have to be aware of as you're starting it. So identifying who is going to be able to pay to have this study go forward. We'd like to think that drug companies will do this out of the kindness of their hearts. They will not. If they do not feel like there is going to be a substantial opportunity for profit, they're not going to be funding these types of clinical trials. And this is an important consideration. A lot of clinical trials are actually using existing drugs. So similar to the horror story that we were talking about with thalidomide, something that was originally a sedative, it was repurposed as a drug to treat morning sickness. That type of thing happens all of the time. Through looking at medical records, through doing literature reviews, you realize, hey, this drug that's being used to treat cancer actually has an impact on cardiovascular health. So maybe we should do a clinical trial to see whether this drug has some benefits to individuals in certain cardiac disease states. That's a perfectly great way to conduct a clinical trial. A lot of the regulatory hurdles are already out of the way. You don't have to worry about what dosages are safe, since that work's already being done, since it's already an FDA-approved medication. Where you are going to run into challenges in those situations are for when drugs are off-label and are in generic form. So if you have a drug that you're now going to prescribe in an off-label setting to examine this clinical condition, but the drug's been around for 50 years treating something else, it's not possible for a pharmacological company to patent that drug. It's already gone into the generic phase. Generic drugs are nowhere near as profitable for pharmaceutical companies. So they're not going to have a whole lot of interest in backing the millions of dollars required to complete this clinical study, even if the drug may be super effective, because it turns out that they wouldn't be able to make as much off of it because there's a generic brand that's already out there. So we'd like to think that clinical trial development doesn't work this way, but it most certainly does. Financial considerations play a huge role. Entities like the National Institutes of Health are able to fund drugs and drug studies where maybe the profitability isn't a big concern there. And that's one of the reasons why entities like the National Institutes of Health are so important in the advancement of clinical trials. They do not have a financial investment in the drugs that are being developed like a pharmacological entity would. They're merely interested in whether this can improve health in the study population. So that's one of the reasons why clinical trials are often funded by those bodies. We've already talked about the regulatory hurdles and the roles that these things can play. Navigating FDA approvals is a very difficult process. It's something that requires expertise in and of itself, knowing what FDA parts apply to your specific situations. Is it a medical device? Is it a life-saving medical device? Is it a drug treatment? Is it an investigational device? Knowing all of those sorts of ins and outs of FDA approval is exceptionally important to ensure that you're able to get a clinical study from start to finish. And here's just some title parts that are related to FDA. And of course, if you're coming up with a solution to a problem, maybe you don't want to limit it to the United States. Oftentimes, we want to have a treatment that can be used worldwide. That requires interactions and conformance with a whole nother entity or multiple entities of approval. So something like the E6 Good Clinical Practice, which is a consolidated guidance created through the for a collection of European countries that include international ethical and scientific standards for conducting clinical trials. So if you do these things, this can be approved. And it covers things like conduct, protocol, informed consent, risk-benefit analyses, validity of the research, quality of the data being collected, record keeping, personnel procedures. So all of the things that we require for a clinical study to be performed correctly are instantiated in this good clinical practice. And many of you may have experienced some of these aspects of clinical research if you have completed your city training for good clinical practice. Now, that's not the E6 good clinical practice requirements, but it's a similar sort of set of materials. It's telling you the types of things that are required for a clinical study to be performed well. And by following those guidelines, the work that you do is going to be of a higher quality, patients will be better protected, and these are all really important aspects. Of course, when you think about the data that you're actually collecting, it becomes really important to have very well-defined standards in place for how you go about doing that. So you want objective data collection as much as possible. Sometimes subjective collections have to take place. Someone has to look at it and make a decision as to whether someone's responding or not responding. But ideally, you want objective data collection that's collected systematically and standardized. So you don't want to have differences in how data is being recorded or how events are being interpreted across investigators. So if you have two people working on a study, you want them to be looking at things exactly the same way. So standardization in those data collection forms. You want to have a data management plan. So you want to have a way to ensure that you are handling, coding, storing, and tracking data and participants consistently. This is important first to ensure that everything's working the way that it's supposed to. It's also exceptionally important from a compliance space. So if you have the FDA show up at your door and say, we want to see your records, that they're able to see step by step how data was collected, how it was analyzed, how it's being interpreted, to ensure that things are being done appropriately. Oftentimes they involve case report forms. So these are forms that are created for the study that are designed to capture all of the necessary information in a consistent, structured format. So everything related to the study makes it into this case report form, and you're going to want to provide that information. You are going to, of course, use validation rules, things to identify missing or inconsistent data. You want to check your data regularly to make sure that it's accurate and complete. So all sorts of data quality assurance has to go on. For data entry and validation, you need to train your staff on proper data entry procedures and use software. Whatever that software may be, people have to know how to interact with it properly. You want to contact or perhaps even conduct double data entry or real-time field validation where information is being entered in multiple times from different parties and then being compared. So if something gets entered in twice and then you compare if it checks out, chances are it was entered incorrectly. You have to look at data security and confidentiality. Databases oftentimes in a clinical study have all sorts of protected health information. You need to ensure that that health information is kept private and entered in consistently, complies with HIPAA requirements to make sure that it's safe. You may have a data monitoring committee, which is similar to a patient monitoring committee, but their specific purpose is to look at the data that's coming in to ensure its accuracy, correctness, and thus in turn ensure participant safety. You may engage in interim analysis and reporting. So as you're conducting the study, maybe you realize something is going to have an impact on your results. You want to make sure that you're reviewing those sorts of things to ensure that you can make changes if need be. Perhaps regularly updating your stakeholders, the people that are funding the study. When it comes to data storage and archiving, you need to store the data in a secure and retrievable manner for a certain amount of time. Oftentimes this may be 3 years following the completion of the study. Some entities require it stored for longer. You want to make sure that it's stored in a safe way and that in the future, if need be, people are going to be able to have access to it. And then, of course, data sharing and transparency. We see the need for this more and more. Once you come up with a result, it's important for you to make that result available to individuals and for you to be entirely transparent with how those data were collected, what was done to ensure reliability and consistency. So as part of that, you want to make sure that you have a way to remove identifying information from your data before you make it available, because of course you can't give away patients' personal information, just the results of the study itself. So in a perfect world, what you should get out of a clinical trial is the conclusion regarding whether the outcome that you're evaluating is positive or negative, how it works. In the real world, it's oftentimes the case that you have to settle, you don't get a complete answer, and then you have to study further. So in terms of steps to launching a clinical trial, firstly, you need to write a clinical research protocol. This is going to talk about the background and significance. It's going to have your questions and hypotheses, have a whole lot of detail about your methods. This is what you start basing your clinical study on. Following that, you are going to provide this sort of information to some sort of regulatory body, and maybe your department, your institutional review board, the hospital or facility that the work is being conducted in. All of these require knowing what it is that you're going to be doing. Now, a couple last things that I want to talk about are clinical trials in COVID-19. So firstly, during COVID-19, we saw the speed and collaboration in developing vaccines progress at a rate that hadn't been seen before. A lot of modifications were made to clinical trial procedures, things like remote monitoring, adaptive trial designs, conditional approvals were put in place to ensure that vaccines could be developed as quickly as possible. So that's really an interesting case study to show how quickly these things can happen if the requirements necessitate it. But it typically takes much, much longer than what we saw with COVID-19, 5, 10, 20 years to go from drug identification to the end of clinical trials. And then the last thing that I want to talk about is sort of the future of clinical trials, and just something to have in the back of your head is this notion of how technological advancements are going to be impacting clinical trials. So the use of artificial intelligence, big data sources, we know is going to result in sort of a boom in personalized medicine. You may be able to provide individualized treatments based on genetic profiles that AI algorithms have identified that if you have these things in place, this is the best treatment for you. Being able to know how to incorporate these technological advances into your clinical trials is going to become more and more important. Similarly, global collaborations. COVID-19 is another great example of this. Researchers from around the world were sharing their data, were sharing their methods, were sharing how they were conducting their research in a global cooperative scale that really hadn't been seen before. But now that we've seen how to do it and we know that it's possible, this is going to become more and more important. So that's just a very quick sort of brief outline as to what a clinical trial is, how you go about doing it, and some specific considerations. And hopefully you've learned a little bit about clinical trials, and it's something that may be of interest to you in the future. Thank you.